Hi, and welcome to the second of two screencasts on blood. I neglected to cover hypoxia when we were talking about red blood cells. I edited the PowerPoints and did not print out a fresh copy because my home printer is kind of wonky. It's not working real well. So, at any rate, we left off talking about anemia, which is an inability of the blood to carry oxygen. Anemia can result in hypoxia, which is reduced oxygen levels in a specific tissue. So, as we said, one cause of hypoxia could be anemia. So, tissues aren't getting oxygen because the blood lacks the ability to carry it. It could also be that a specific tissue is experiencing hypoxia because the blood flow to that tissue has been cut off. So you lean over the back of a chair, like with your arm over the back of a chair, and put pressure on your axillary artery, and then your arm falls asleep. That's hypoxia. Or it might be you're climbing a mountain, you're at altitude, the air is thinner, there's nothing wrong with your blood, but there's just not as much oxygen available in the environment, so you are hypoxic even though there's nothing wrong with your blood. So make sure you can keep straight the difference between hypoxia and anemia. Anemia generally refers to something is wrong with the blood, and hypoxia just means there's a tissue that's not getting oxygen. Now we get to the meat of our lecture, which is our white blood cells. What you need to know is that there are two different kinds of white blood cells, or groups of white blood cells. The granulocytes, which are your neutrophils, eosinophils, and basophils, and then your agranulocytes, which are your lymphocytes and monocytes. They are clumped into these two categories because the granulocytes have chunks of stuff, cytoplasmic inclusions, that we call granules that show up as specific colors when you stain the blood in a specific way. So all of these fills are named for the type of stain that their granules like to absorb. So fill is like philic, it means liking. And then the agranulocytes lack the granules, and they are the lymphocytes and the monocytes. So your white blood cells make up only about 1% of your total volume. They have the ability to crawl out of your blood vessels. So this is called diapedesis. And you don't want to think of it as your blood is under pressure and oops, a blood cell popped out of a capillary. This is a multi-step process whereby the cell has to attach itself to the wall of the blood vessel, physically crawl out of the blood vessel, which may involve making endothelial cells let go of each other, and then they let go of the blood vessel and enter surrounding tissues. So they actually have to locomote and be able to grab onto blood vessel walls and move around. So it's a very specialized skill. Leukocytosis just refers to an elevated white blood cell count. And you're going to see later on that this is part of your body's normal inflammatory response. Whether you've got a bacterial infection, a viral infection, or maybe you've just cut yourself and it's not infected, you still end up with some inflammation. And leukocytosis is part of that. So the first granulocyte that we are going to talk about is the neutrophils. They are so named because there is a neutral pH stain that the granules in a neutrophil tend to absorb. So that's where the name neutrophil comes from. They like the neutral stain. In our lab, they are not going to have this reddish purple, very small dots in them. They are going to look like they have nothing in their cytoplasm. So they're going to be this funky shaped nucleus with clear purple cytoplasm. Because of the funky shaped nucleus, they are sometimes also called polymorphonuclear nucleocytes, which means many different shaped nucleus. The important thing to know about them is that their granules contain hydrolytic enzymes called defensins that allow them to kill bacteria. So neutrophils kill engulf, and phagocytize bacteria. That is their primary job. The next of the granulocytes is the eosinophil. They are so named because they absorb this red stain called eosin, which is named after Eos, the Greek god of the sun. So now you're not only learning about blood, but Greek mythology. What could be more interesting? So the primary job of the eosinophils is to digest parasitic roundworms, which you generally don't think much about, but we're all covered by roundworms, and occasionally they end up in the bloodstream. Your eosinophils go after them. Last up is the basophils. They are so named because their granules like to absorb a stain that I believe is called basic fuchsin. So it is a basic pH stain that is purple, and hence their cytoplasm stains with these dark purple dots, which are the basophil granules. These granules contain histamines. If you've ever had allergies, you might notice that you're taking drugs called antihistamines. What the antihistamines do is block the inflammatory effect that histamines have. Keep in mind that if you have an infection or a possible infection, inflammation is actually a good thing. We like to keep it under control when there isn't an actual infection, and then it's kind of an impediment to healing. But most of the time, a little bit of inflammation is a good thing because it helps to fight an infection. So those are your granulocytes. Then you have the agranulocytes. First up is the lymphocyte, known because they spend a lot of their time hanging out in lymphatic tissue, which we will talk about in lecture. So they can be found in the lymph. They also circulate throughout your bloodstream, and they are the primary cellular component of your immune system. There are two different types of lymphocytes, T lymphocytes and B lymphocytes. We'll discuss both of them in much more detail when we go over the immune system. For now, know that your T cells are responsible for taking out virally infected cells. So one of your cells becomes infected with a virus, it is now a liability. The T cell assassinates it. It's pretty cool. And then the B cells are the cells in your immune system that produce your antibodies. And we will just leave that at that. Next agranulocyte is the monocyte. This is the biggest of all of your formed elements. It's this great big cell with usually this kind of almost puffy M-shaped nucleus. What they do is crawl out of the bloodstream, diapedesis, and enter into your tissues, and then differentiate into different kinds of macrophages. So macrophages are a type of phagocyte. Right? A phagocyte is anything that can phagocytose things, which is cell eating. Macrophages just means big eaters. So they are phagocytes with, I think, what the book describes as a prodigious appetite. And their job is to phagocytose anything that's not supposed to be there. A virally infected cell, some of your own dead cells, debris, bacteria, whatever it is, monocytes clean it up. And that's what they look like. So white blood cell production also takes place in the bone marrow. So all of hematopoiesis, erythropoiesis, and leukopoiesis takes place in the bone marrow. There are two different types of chemicals that direct the formation of our 5 different white blood cells. They are the interleukins and the colony stimulating factors. Do not worry about numbers and names of them, just know interleukins and colony stimulating factors. Things get real crazy real quick when you start trying to track down all of the interleukins because your immune system is ridiculously complicated but also interesting. So there are some leukocyte disorders that you want to be familiar with. The first up is leukopenia, which just means a low white blood cell count, and then leukemia is cancer of your white blood cells. So it means that your white blood cells are dividing faster than they're supposed to be, And this ends up being bad because not only do they divide faster than they're supposed to be, they lose the ability to do the things that they're supposed to do. So your bloodstream becomes clogged with all of these leukemia cells that are getting in the way of all the other cells and not doing what they're supposed to do, and reducing the numbers of healthy functional white blood cells that you have. This then leads us to platelets. Platelets, we said, were fragments. So here you have this megakaryocyte. The megakaryocyte stays in the bone marrow, and then it just sheds little pieces of itself, like really big vesicles, instead of coming into the cell, blob off the outside of the cell, make their way into the bloodstream, and that's what makes up the platelets. The hormone that controls the production of platelets is called thrombopoietin, which is why platelets are sometimes called thrombocytes. The job of a platelet is to form a plug whenever a blood vessel breaks, so that the blood vessel stops leaking until such time as you can get the formation of a clot, which is physically and chemically a different thing. So this is one of the other open-ended essay type questions in the chapter called vessel healing. There are 3 steps to it. The first, as it says here, is vascular spasm. Smooth muscle will contract, causing vasoconstriction. So the smooth muscle in the tunica media constricts, shuts down or reduces blood flow through that wounded vessel so you don't get as much bleeding out. Then the platelets are going to stick to the exposed connective tissue in the tunica media and tunica externa. They stick to that. They release platelet attracting factors, which attract other platelets, and you get the formation of a plug. You might remember this from AP1 as an example of positive feedback. And then the third step is coagulation. So keep straight, blood vessel healing is spasm, platelet plug formation, then coagulation. We are going to treat coagulation as a separate topic. You can continue to look at that last slide, or think about what's on that last slide, I should say. And I'm going to show you the parts of this slide that you need to know. So on the last slide, we talked about the intrinsic pathway and the extrinsic pathway. Intrinsic just means that the signal for coagulation is coming from the platelets. So when the platelets stick to the connective tissue, they release a chemical or protein called platelet factor 3. Platelet factor 3 leads to the production of this thing called prothrombin activator, and that's where coagulation starts. The extrinsic pathway is when cells get busted open. Stuff that's supposed to stay inside a cell ends up outside the cell. One of those things that comes out of a busted open cell is tissue factor. So this tissue factor gets released. That leads to the production of prothrombin activator. After prothrombin activator is formed, we'll go over the text on the last slide. Here you have it. Prothrombin activator catalyzes the conversion of prothrombin to thrombin. Thrombin is an active enzyme. Prothrombin is an inactive enzyme. So it gets activated by prothrombin activator. And then the job of prothrombin is to catalyze the conversion of fibrinogen into fibrin. Fibrinogen is a soluble protein that floats around freely in the bloodstream. Fibrin is insoluble. It becomes unfolded and sticky. One fibrin molecule will stick to another fibrin molecule, and you get this big fibrin mesh. So this used to be an open-ended essay question. It is not an essay question anymore. So you just need to know the basic definition of what does prothrombin activator do, what are all of the things on here, and what is the intrinsic pathway? It's PF3. What is the extrinsic pathway? It's tissue factor. And that brings to a close blood with about 20 seconds left to spare, because I can only make 15-minute movies for free. Thanks for listening to the end. I hope you're enjoying your spring break.